we move to the 2014 David Craig Medal, which honours outstanding contributions to chemistry by an Academy Fellow. And of course, it's in honour of Professor David Craig, himself a most eminent chemist. The recipient of the 2014 David Craig Medal is Emeritus Professor Kurt Wentrup, and I invite Kurt to come to the stage.
Thank you very much indeed.
Yeah.
Thank
you. Numerous chemical reactions take place via reactive intermediates, that is, short-lived molecules that are undetectable under ordinary reaction conditions. In order to understand chemical reactions, it is necessary to understand the role of these reactive intermediates. Professor Wentrup has pioneered methods to study them and observe them directly by combining the technique of flash vacuum thermolysis with low-temperature spectroscopy. The resulting knowledge is of fundamental importance for theoretical chemistry as well as practical applications in the synthesis of new types of compounds. Professor Wentrup, my congratulations. I now invite you to address the meeting on short-lived molecules, research on reactive intermediates, and chemical reactivity.
Thank you very much indeed, ladies and gentlemen. It is, of course, a great pleasure and honor for me to stand here today and to receive this reward, which I hope that I can live up to, and also to present you a very short story on our work on reactive intermediates and unusual molecules. Reactive intermediates are short-lived molecules that occur as transients in the course of chemical reactions. Because they're transient, they're difficult to catch and to observe and to characterize. One has to use special methods to do this, and I will show you some of the methods that we use. Why is it important to study these compounds? Because they are so difficult. It is important in order to understand the way that chemical reactions take place. And once one understands that, then one can use that knowledge to predict new reactions. For example, the one shown here, just a small section of the kind of chemistry that we have done recently. For example, you can make psychotropic drugs from this kind of reactive intermediates here, which I will show you how we make these. And you can make another important class of compounds, antibiotics, such as quinolones and naphthyridones. I will show you here a short summary of the kinds of molecules that we have been interested in recent years. The molecules just mentioned before are these long-chain cumulines, in this case with 4 cumulated double bonds. One might think that such compounds are very strange and unstable, but in fact it turns out that some of them are reasonably stable. That means you can observe them in some cases even at room temperature. In other cases, you have to go to very low temperatures, and I will show you how we do that. Generally, it turns out that cumulines like this with an odd number of heavy atoms are relatively stable. Think of CO2. It is, of course, an odd-numbered heterocumuline, and it is very stable. In contrast, those with an even number of heavy atoms, like C2S2, is extremely unstable. Nonetheless, we can make this compound and characterize it at low temperatures, and I will show you how we do that. We're also interested in Twitter ions of various kinds, like N2S. N2S is the sulfur analog of laughing gas. Laughing gas is a very stable compound, but N2S is extremely unstable. And again, we need to use special methods to observe and characterize such compounds. Nitrile imines, nitrile ylides, and so on. Carbenes and nitrines are compounds we have worked a lot with, but I won't have time to tell you about all this. Here's N2S. The story starts in the 1960s, actually, with the discovery that thiatriazoles, this kind of molecule, decompose thermally with loss of nitrogen and formation of alkyl cyanates. This was, in fact, the first synthesis ever of alkyl cyanates, which are themselves unstable molecules. And so-called sulfur in quotation marks, because it was assumed to be sulfur, We found out that, in fact, the reaction is not as simple as that. It is a cleavage reaction. The molecule cleaves here to generate the cyanate and N2S. So N2S is, in fact, the first sulfur species that is formed in this reaction. The evidence that we have is the low temperature infrared spectrum that you see here. The strongest band in the spectrum is in fact due to N2S, which can be demonstrated in various ways. We have also obtained the mass spectrum and the photoelectron spectrum of this compound. The other strong band in the spectrum is due to the cyanate, which is necessarily formed as well. This diagram shows you some of the apparatus that we use for this kind of investigation. We do need to use special apparatus, and we have developed this over a number of years. And the top diagram shows you a flash vacuum pyrolysis or thermolysis apparatus, where the action takes place in this quartz tube here, which is about 10 centimeters long and 1 centimeter diameter. It's all in high vacuum, 10 to minus 3, 10 to minus 4 millibar. And the molecules that are formed here by pyrolysis in the hot zone only experience a few collisions with the hot wall because of the high vacuum and the short contact times of the order of milliseconds. And therefore, we need high temperatures for anything to happen at all. If we use temperatures of 200 or 300 degrees centigrade, nothing would happen because the molecules simply only make a few collisions with the hot wall and only pick up a small amount of energy. Therefore, in general, we need high temperatures of the order of 500, 600, 700, 800 degrees. These are simply nominal temperatures because we need to give the molecule some energy so that it can react and generate these reactive intermediates. Immediately when they exit from the pyrolysis tube here, they're deposited at this point at liquid nitrogen temperature, which is 77 Kelvin, because this target here, which is usually a KBR window, is cooled with liquid nitrogen through these dewars. And the whole system is pumped through this chimney here. So that way we can isolate molecules and observe them spectroscopically at low temperature. At the bottom, we have the same apparatus here, the same pyrolysis oven, attached directly to the ion source of a mass spectrometer, which operates always in a very high vacuum. So in this case, immediately after exiting from the hot tube here, the products that are formed are ionized in the mass spectrometer and can then be recorded. This is an example of C2S2. That is a short-chain cumulene and therefore very unstable. It is, however, thermodynamically stable enough that we can generate it at high temperatures in a flash vacuum thermolysis reaction. I won't go into detail with all the chemistry here, but the diagram here, or spectrum, that's an infrared spectrum, recorded at 10 Kelvin actually. In this case, we have to go to very low temperatures, 10 Kelvin, in order to observe that compound. In the apparatus that I showed you before here with liquid nitrogen temperature, we wouldn't see anything whatsoever. The compound is not stable enough for that. But here at 10 Kelvin, we see, in fact, the band in the middle of the spectrum is due to C2S2, this compound here. And you can also even see the isotope satellites here due to S34 and C13, which help to characterize the molecule. The other two peaks of importance in this spectrum are due to ethylene, which is a necessary byproduct formed in this reaction. In the diagram at the bottom, this comes from the mass spectrum. You see that this ion here, that is the ion of 88, which is in fact due to C2S2, keeps rising all the time. Essentially the compound is formed in quantitative yield. This is a very clean reaction, forming a highly unstable molecule in a clean reaction. For that, we need to go to 10 Kelvin, as I mentioned, and for that we use an apparatus that looks like this. So this is a liquid helium cryostat, where you reach a temperature down to 7 Kelvin, in fact, at the center of this cavity. Here's the pyrolysis oven again, same ones that I showed before. The pyrolysis takes place in here in a quartz tube in high vacuum. The compound is deposited here together with argon at 7 to 10 Kelvin. 10 Kelvin is good enough in most cases. And then they can be observed spectroscopically using infrared, UV, electron spin resonance spectroscopy in most cases. This shows a diagram of the same apparatus. So here's the hot tube, and here's the target where the molecules are condensed after flight time of a few centimeters. Finally, the aminopropadienones, these are the long-chain cumulines that I started out with. We can make them in various ways from these various precursors. I won't go into details with the chemistry. We can observe them spectroscopically. Here's the example of the smallest member of this series, the methyl derivative, which is a very unstable compound. But larger members with larger R groups here are, in fact, in some cases, as I mentioned early on, even isolable at room temperature, as long as you keep nucleophiles away from them, because they are highly reactive with nucleophiles, and that is important synthetically. Nucleophiles will react with this carbon atom here, which is a carbonyl carbon, and this carbon atom here, which is an imine carbon. So these compounds are thermodynamically stable, but extremely reactive, and therefore, again, they are difficult to catch. What is important is that if we don't want to observe it spectroscopically, like here, using flash vacuum thermolysis, we can also use microwave irradiation in solution. And that's a very easy and simple method to prepare compounds. So we do the reaction in a microwave oven, a little more fanciful than an ordinary kitchen microwave oven, but this principle is the same. And that generates the reactive intermediate. We cannot observe it under those conditions, but we can do chemistry with it. So, for example, we can add nucleophiles, like bis nucleophiles, and we can make cyclic compounds like diazepinones and benzodiazepinones in particular. All these compounds are important for their biological properties. Many pharmaceuticals are, in fact, the benzodiazepinones. They're well known for their pharmaceutical, in particular, psychotropic properties. So here again are some examples of compounds we can make from these intermediates by doing microwave irradiation in solution, trapping them as they are formed. Also, I mentioned the quinolones, naphthyridones. We can also make the thioanalogs, which is a new class of compounds. And with that, thank you very much for your attention.
Thank you, Professor Winthrop. And are there any questions or comments? Please go ahead. Andrew. Please identify yourself and your institution.
Andrew Holmes, University of Melbourne and CSIRO. Kurt, thank you very much. You alluded to the fact that some of these processes can be carried out in solution, and we have done some similar reactions in continuous flow. Do you think this can be scaled up to a manufacturing process by doing it in flow on a larger scale?
Yes, I do, because, of course, flow chemistry is something that's very fashionable nowadays, and in principle, with microwave irradiation, you can flow through the microwave cavity, and so you can do the process continuously on any time scale that you want. And even the flow chemistry is being taken up in industry as a very attractive method. You can do flow photochemistry, you can do flow thermolysis in this manner. And also I should mention in this regard that flash vacuum thermolysis, which is our daily bread, so to speak, of course, this we do usually on a small scale, but it can be scaled up too. It's only a question of the size of the apparatus really and the time, because it takes time to sublime the material through. But actually there are industrial processes that are based on this principle. They don't usually do it in high vacuum, but in low vacuum or in the presence of a carrier gas, so you can make kiloton properties, quantities of compounds, even by so-called flash pyrolysis.
Are there any further questions or comments? Mr. Winthrop, I have a question for you. What is the time scale of this formation of these intermediate, and how long they last, and to be able to capture them?
Right. The time scale, that is the residence time for a molecule in the hot zone of the apparatus I showed, is of the order of 1 millisecond. So that is the time the molecule is on the fly, so to speak, before it is isolated at low temperature. Then it lives forever. If the temperature is low enough, it lives forever. Like, think of interstellar space, where there's a very low temperature and high vacuum. Very short-lived molecules live. They're not short-lived under those conditions. They live forever until they bounce into something and react. So the same in our low-temperature matrices. When the temperature is low enough, the molecules live in principle forever, so you can observe them at leisure. It often takes days to do this kind of experiment. That's because of the instrumentation. But the molecule that is formed in the gas phase, in the hot zone, really only experiences a very short period of time.
On that note, could you please join me to thank Professor Winthrop?